doesn't work once, well then redo it. If it doesn't work twice, well now it's time to troubleshoot. And so thankfully today my experiment finally worked, but troubleshooting is a huge part of science, and actually like one of the biggest things about grad school is learning to troubleshoot and optimize. So today I just wanted to go over some tips for troubleshooting. Starting with, don't troubleshoot if you don't need to troubleshoot. So this sounds like a lazy cop-out, but a lot of times an experiment, when it doesn't work, it's just because of some like silly error, some human error. Maybe you didn't get the liquid out of the pipe, the liquid got stuck in the pipette tip or something, which is why you should always be watching it go up and down. But the bottom line is that when an experiment doesn't work, especially if it's something that is not expensive or time-consuming to redo, the simplest thing is just to redo it and see if it works. A lot of the time it will, and you never figure out what went wrong, but you can make yourself go like bananas if you're trying to like troubleshoot every single item. I've seen this happen to myself, to other people in the lab, where we can have like 20-minute long discussions in group meeting trying to figure out what went wrong, and then we just end up like, just redo it, and then it works. So don't feel like you need to be doing the super scientific thing and troubleshooting every single step. If something doesn't work the first time, the simplest thing is to do it again. This is the advice that I give people. If the problem repeats, then it's really time to get down into this nitty-gritty troubleshooting. So when you're doing that, the super duper duper important thing I'm always stressing is to know why you're doing everything. And when it comes to troubleshooting, well, now it's really important, because if you don't know what each step does, well then how do you know like where something went wrong? So you want to like listen to your experiment when it's talking to you. So listen to your results. They're going to point, not all like failures of experiments are the same thing, unless it's something where it's like you get a signal or you don't get a signal. You maybe get one signal where you're expecting a different signal or something like that. Typically there's some sort of clue about what is going wrong in your experiment. And so you need to know then, okay, well, if I got this result, what step would that have been a problem with? When you are redoing that experiment, even if it's just one of those things where like you just simple redo, you want to, if possible, include more controls, include like, so negative controls, things that shouldn't have a signal if there's like nothing there, and positive controls, things that should give you a certain signal if there's something there. So for example, if you are doing a plating for a bacterial selection, and you have an antibiotics in your plate, you should have a plasmid with that antibiotic as your positive control, that then this plasmid will be able to grow in these bacteria, and so then hopefully if yours has the plasmid with the antibiotic resistance, it should be able to grow. So that would be your positive control. But, well, what if it's growing just because anything can grow? So now you need a negative control where you don't put any bacteria on there, and then that shouldn't grow, or you put bacteria that have a different antibiotic resistance gene. And so you can see that there are different things that can tell you different things about where things might be going wrong in a process. If you don't get any colonies, if you get like a lawn of colonies, these various things, they're telling you different things, and by having these controls, you can now know what step might have been wrong. If things are like a really long-winded protocol, you shouldn't wait until like the very end. As long as it's feasible, don't wait till the very end to get some sort of readout. So you should be doing some sort of checks along the way, even if you're not testing them directly. Say you're doing a protein purification. Here what you want to do is you want to keep samples all along the way. So keep a sample so you take a cell and then you like break the cell open, so you lyse it, and then you're going to purify the protein out of the cell, say. Well, your protein, if you're expressing a soluble protein, it should be in that soluble part. It should be in that liquid. But what if it is misfolded and it ends up in the pellet? Well, now if you've tossed your pellet all the way in the beginning, you don't have a way to find out. But if you keep that pellet, keep a sample of the pellet, run an SDS page of it when you're running things at the end. So sometimes I would just keep samples along the way. So keep a sample of the pellet, keep a sample of the lysate, keep a sample of like all of these various steps along the way. Keep a sample of the flow through if you're doing some sort of column where it's supposed to bind the column, well maybe it doesn't bind the column. So keep all that stuff along the way. If you you probably won't need it, and you probably won't even need to like run a gel on it or something. But if something is wrong at the end, then you have those samples you can go back to, and you can look and see where things might have gone wrong. So it's always good to keep those samples along the way of all these different steps, so that you can then go and look and see where something might have gone wrong. And then if you know how that step works, then you might be able to troubleshoot based on that. Sometimes if an experiment is not working and it really, really should work, like you know this should work, then you might want to try checking and making sure that all your buffers are okay. Like just remake all the buffers if it's buffers that you made yourself. I had this problem once. I never really figured out exactly what the problem was, but there was something wrong with one of my buffers and it was just messing things up. I never even figured out which buffer it was, because it was one of those, or it might not have even been a buffer, but one of the buffer components. It was one of these experiments where there were like 20 different things in this buffer, and I could have gone through and like remade one at a time and done this experiment over and over. But the simplest thing there was just like start from scratch with all of these reagents, all these different chemicals, and then make it, and voila, it worked. So I kept all those other stuff in like a bag, like old stuff. So if I really wanted to, I could have like gone and tested, but it just wasn't worth it. So you need to figure out like what's worth your time and energy, and so that's why sometimes just like redoing things is worth your time and energy. But yeah, so if something's not working and you know it really, really should work, then the buffers might be a problem. You might have some contamination in the buffer. Your pH, check your buffers. Maybe the pH is really off. This happened to me once. I don't know how it happened, but the pH was like way off. Yeah, so check those sorts of things. Then there are going to be times when something isn't working, but you don't know that it should work. So basically, when you're trying to modify someone else's protocol to work on something different, or you're trying to develop a whole new protocol. That's one of the most awesome things about biochemistry, is that you get to be really creative, and you get to design experiments to test things that you want to test, and not just follow recipes in a book. But the problem is that because you're not just following recipes, well, then you have to like figure out how to make things work the way you want them to work. And so there's going to be a lot of troubleshooting, but also like optimizing. So Troub-optimizing, we're going with it. Okay, so there's going to be a lot of Troub-optimizing. That's going to be like a huge, huge part of being a scientist, is Troub-optimizing. And so you're going to then need to really know what each step is doing and why you're doing each thing. That's one of the reasons why I keep drilling it into you. So then you can once you have these basic skills down, you can mix and match them in different ways and design your own protocols. But a lot of the time, that's not going to work very well. And so you have to just kind of try different things then. And by knowing what all these different steps are doing, you can then figure out how to change different things, and by keeping samples along the way so that you can test different things. If you're trying to do something like for the first time, if it's something that you're trying to do from a sample, try if possible to make extra. So if you have, say, a cell lysate or something, or you have even save extra cell pellets. So you say you grow some protein in cells or whatever. Try to keep the like an extra cell pellet, like make enough, like freeze down so that you have a backup that you can try again. Or if when you break those cells open, keep parts so that you can try again. So it's always good to have like a little stock set aside so that you can try the same thing with the same sample if something goes wrong along the way, and then you don't have to go back to the very, very, very beginning. And so yeah, but then along the way with all your experiments, you want to be taking samples of things that you can test for, like test for things later. So keep the flow throughs, keep all of this stuff, especially when it's the first time you're doing an experiment. And if something doesn't work the first time, then try it again, but but if possible, include more controls. And so a lot of times we kind of get lazy with our controls. Especially if it's the first time you're doing something, you really, really, really want to make sure you have those controls. But if you're doing something a lot, and then it just like randomly doesn't work, it's probably just some human error thing. But you want to you be really sure that when you do it, to redo it, you're including controls if possible. So your negative controls, make sure that you're not seeing a signal if you shouldn't see a signal. Your positive control, make sure that you see a signal if you should see a signal and that the signal looks like it should be. That sort of thing. Yeah, sometimes there aren't really good controls, especially when you're trying to develop a protocol or where you're trying to do something where you're purifying a protein or something and you don't have any of that protein to compare, and you're like, I'm trying to get the stuff to later use as a control for some other experiment. So there's not always like a control. That's why you need to be able to then know how to interpret different things about different results. And so much more on the posts are all the different techniques of things like, okay, well, if you're running a PCR and you see multiple bands, what are the things that you can do? If you're running an SDS page and you see some weird smear, what are the things that you can do? And so really, so check out the details of all of these different experiments. I'm not going to go into all of them here. And yeah, this was just kind of like a rambly post, but people had asked me, and I get asked a lot about various troubleshooting things. And the first question sometimes is well, have you done it twice? And sometimes it just works the second time. And you never figure out why, but it saves you a lot of time and energy that you could have been trying to figure out why. Especially because sometimes the stuff's already thrown away and you don't know. Yeah. But anyway, troubleshooting is, so sometimes that works, that doesn't always work. And if it doesn't always work, then it's time to troubleshoot. The thing is there's going to always be more things that you could test, and testing every component when you're troubleshooting isn't really practical a lot of times or sensible, which is why you should know what each step does. But sometimes even if you do, there could be a couple different things that you think might be going on, so then you could test those different things if it's something that's cheap and easy and you have enough sample. Sometimes the problem can be where an experiment is really tedious, it's really long, you have limited sample, it's really expensive, something like this, where then you have to be able to choose, okay, what do I think is most likely to be wrong? And hopefully there are things that you could do to try to test before you jump in. But you know how they say okay, we'll try to minimize how many variables you change at once so that you know if the variable you changed is a thing. But sometimes you just you have to choose and you have to change multiple things at once. And so that's why it's really important to know what you're doing in your experiments and where things might be wrong. Try to do those little tests to see on small scale what is wrong before you try and do things larger scale. But do know that sometimes you're gonna have to try change multiple things at once, which is against the knowing what was actually the thing. But it can be what you have to do, and ultimately hopefully it works. And be ready to troubleshoot. Know that that's going to be a huge part of being a scientist. Don't feel bad about it. Know that experiments often don't work, especially the first time, especially if you're trying to develop a new protocol. And know that that's part of being a scientist, and that to expect that, and don't feel bad about yourself. Other people around you, they've been through the same thing. They know it well. And so yeah, be ready for that and be up for the challenge of trying to get things to work when they don't want to work, and then be really proud when things do work. Oh, and also at a group meeting and stuff, or if you give a poster presentation, don't worry about if you don't have really good results or whatever. I have to give presentation, a poster presentation and a group meeting and stuff. It's I've been troubleshooting for months, but that sort of thing, it can be really helpful too to talk with other people in your lab and to even present these partial results that then people can help you interpret. Maybe they can help you, they have ideas to help you troubleshoot, to help you optimize in these various things. So don't feel bad if your experiments aren't working. Know that progress can be slow at times, but then once you get something working, then it's boom, boom, boom. That happened to me in grad school. I had this big experimental problem, and then I finally figured it out and I got a lot done. But I felt really bad about myself before then, but don't judge yourself based on your experimental results. So yeah, so be nice to yourself, and yeah, also take time for yourself. I know I'm not a great example of work-life balance, but I really love science. I love being in the lab. I love teaching people, but yeah, so be nice to yourself, and know that troubleshooting is something to expect, and Troub-optimize, the best of them. So hope that was at least helpful in terms of just getting you prepared, or the sense of mind. I don't know.